Welcome to all of our viewers and listeners for another Road to Wellness webinar. I'm Kathy Olewski, and I'm the Road to Wellness host, and I bring the patient perspective to this webinar series. I'm in my 8th year in remission off treatment with ANCA-MPA, and I hope to help the Vasculitis Foundation bring awareness to our rare diseases. Today, we have a COVID-19 update webinar about vaccine efficacy for patients with vasculitis. We've gathered questions from our vasculitis community, and our guests today will help us by answering those latest concerns. So let me begin today's webinar by introducing our guests. We have Dr. Anisha Dua, who is the director of Northwestern Vasculitis Center. She's an associate professor of medicine and rheumatology and the director of the fellowship program at Northwestern University Feinberg School of Medicine in Chicago, Illinois. We also have Dr. Kevin Byram, and he's the director of the Vanderbilt Vasculitis Clinic. He's an assistant professor of medicine in the Division of Rheumatology at Vanderbilt University School of Medicine in Nashville, Tennessee. And we also have Dr. Sebastian Satui, who is the director of the University of Pittsburgh Medical Center's Vasculitis Center. He's an assistant professor of medicine at the University of Pittsburgh, and Dr. Satui was awarded the 2019 to 2021 Vasculitis Clinical Research Consortium's Vasculitis Foundation Fellowship Award. Welcome to all of you. We totally appreciate your time here today, and we're excited to get started on our webinar. And I think with that, I'll begin by just turning it over to Dr. Byram and let you move forward with educating us first.
Sure. Thanks, Kathy. I know on behalf of the other guests here, it's always a pleasure. So I thought we would start out by just doing 8 to 10 minutes kind of going over antibodies and how it relates to rheumatic disease, and of course, in particular, systemic vasculitis, particularly in the context of the global pandemic. This will give us a good framework to kind of build upon and jump off of as we enter into question and answer. So let's see here. I don't have any disclosures. We're going to look at the purpose of our immune system and explore some of the ways in which it works. And in one of those ways that it works is antibodies. Of course, some of the medicines we use in rheumatology and in the care of patients with vasculitis affect antibody function. We'll go over that. And then we'll talk briefly about vaccines. And of course, all of this is through the lens of COVID-19. The purpose of our immune system, this is how I talk to my patients about this. Our immune system is a great thing. It protects our bodies from the outside. This is bacteria, virus, fungi, mycobacteria like tuberculosis, but it also protects us from ourselves in some sense. Our immune system is playing a constant game of Goldilocks, trying to be just active enough, not too overactive, not too underactive. Our bodies produce cells that it's not supposed to sometimes. This is what we call cancer, and our immune system is very good at detecting that and not letting it become a problem. On the flip side of it, as our patients know, the immune system can get too active, and that's what we call autoimmunity. So our immune system is at any given time a very complex system that is protecting us both from the outside and the inside. And the way that our immune system does that is highly complicated. I tend to draw an analogy when I'm talking to my patients about like the armed forces or a country's armed defenses. They might have a Navy, an Army, an Air Force, a Coast Guard, and our immune system is very similar in that we have different groups of cells that do different functions and protect us from different organisms. Today, we're going to be focusing a lot on B cells and T cells. These are what we call lymphocytes, but it's very important to realize that our body has many different types of immune cells. For example, we have eosinophils, which patients with EGPA might know well, and these are allergic type cells. And then we also have other things like neutrophils, which are over here, which are almost like the first initial foot soldiers of any kind of immune response, particularly bacterial. So I just wanted to lay this out there that there's a lot of different ways that our immune system is working at any given time. We're going to be focusing a lot on B cells, and because that's where antibodies are made. These are antibody factories. And so what is an antibody? These are proteins. In cartoons, they're usually drawn as a Y-shaped molecule, but they can connect together in different ways and become a little bit bigger depending on what the molecule is. But it's really important to know that these are really complex molecules. This down here in the bottom right corner is kind of a protein folding drawing showing just how complex a single antibody is. And there's a lot of different variations. At any given time, we have thousands, 10,000s, hundreds of thousands, probably millions of different antibodies floating around that are primed to recognize different things that aren't supposed to be there. They can exist alone. Many times they do, just floating around in our serum or in our blood, but sometimes they're connected to the actual B cell, and these are called B cell receptors, if that's the case. But these function, in a cartoon we'll look at in a minute, to neutralize foreign invaders, or, as we described, even some cells that we produce that aren't supposed to be there. So how does an antibody work? In this cartoon here, the red things are bad, right? Red is bad. And these are, we'll call them bacteria here. The blue Y-shaped molecules are antibodies. And antibodies can work in a couple of different ways. They can mark a bacteria for other cells to come and activate. They can surround a cell and start killing processes themselves. But they can also do what's called agglutination, which kind of sticks all the bacteria together and makes it really easy for when a molecule does come to help out, all the bacteria is sitting there waiting for it to be acted upon. In this cartoon, the yellow cell here is a good cell. It's a cell that recognizes that antibody and says, this thing that's connected to this antibody doesn't need to be here. I'm going to kill and process it and destroy it. And so that's typically how an antibody works. It's important to realize there are kind of different types of antibodies. I will have a point here in a second that early on in any kind of infection, there's a really high, well, let me orient the viewer here, the listener. We have days on the x-axis since infection, and then we have quantity of the antibody or DNA or RNA as it may be on the y-axis. Early on in an infection, you can detect a lot of live virus and a lot of viral genetic material. About a week into it, you'll start producing the first antibody, which is called an IgM. And then about a week later, you start producing IgG, and that is what kind of takes over as the body kind of gets rid of or stops shedding viral RNA and really stops producing the IgM antibody. This is important because of the different tests that we'll talk about in a minute with respect to COVID. Most antibody tests are IgG, and COVID-19 is no different. There are some infections that we can detect with an IgM. We're just not using that in COVID as it stands. Generally speaking, early on in the infection, we're using the viral genetic material as the diagnostic to identify early infection. So how do antibodies work and vaccines work? These, most vaccines, kind of in the general knowledge, vaccines train your immune system to produce antibodies. There are a couple of ways to get an antibody to a certain pathogen. One is by dumb luck. That does not happen too often, though our bodies are primed to kind of make antibodies to very, very common things, toxins that bacteria produce and things like that. But these viruses are pretty complex. So really there's only two ways to get antibodies to them. One is to get the infection itself. You go through all of that we just looked at. You have viral genetic material, you produce an IgM, and ultimately you produce an IgG that hopefully protects you from the pathogen. But the other way to get immune is to get a vaccine, and it kind of uses your body's immune machinery to just start producing those IgG antibodies to protect you against the pathogen. This is called the B cell response. Now, what will become very important as we jump into the questions here is that vaccines can work in other ways. I showed you all the different actors and arms of the defense of the immune system, and that was for a purpose, and really to show you that vaccines can work in other ways. In particular, there's what's called a T cell response. And this is not effectively measured by antibody level. And just to give you some scope as well, there are many different kinds of T cells. There are T cells that kill things. Those are called effector T cells. There are helper T cells, follicular helper T cells that kind of activate other T cells to kill. So this T cell response is really complex and not effectively measured by kind of general laboratory techniques. It's also important to understand there are two different types of antibodies with COVID-19 that are available from a lab testing standpoint. The one early on that everyone heard about was the nucleocapsid antibody. This was before there was vaccines, and this is the one that identified past infection. The spike antibody, that's the protein that the vaccines are making, the spike antibody. So that could be, if that level is detectable, that could be a signal of B-cell vaccine response. But the truth is that could be a indication of past infection as well. So sometimes it takes both antibodies to understand if the infection has been there or not. And it's just important to understand that the PCR testing that is done, the nasal swabs and things, are not antibody tests in the general sense here. Those are looking for that viral genetic material. This will be the meat of the discussion as we get into the question and answer, but we of course use a lot of different medicines in rheumatology that by design suppress the immune system. One of those approaches are therapies like Rituximab that by definition deplete B cells. Multiple sclerosis patients have another one called Ocrevus, which is another B cell depleting therapy, but these by definition decrease the number and the function of the antibody factories, those B cells. This helps us treat diseases like ANCA vasculitis because ANCA is an antibody. We're wanting to decrease the number of cells that are producing that antibody that causes problems. But that comes with a cost, and that really, those antibody factories do good things for us too. Now, it's not, there's a high complexity to this, and we have memory cells that can help us remember vaccines we've gotten in the past. But Rituximab really targets kind of these B cells before they get to the memory phase. This does make it potentially a drug that can decrease the effectiveness of the B cell responsive vaccines. It's important to know that we have other medicines, glucocorticoids and methotrexate are the biggest ones that have the most evidence, I should say, that also can impact vaccine response. And those mechanisms are not entirely understood, but likely deal with some of the B cell response as well as the T cell response. I think this is a good time for me to probably pass it to Anisha and direct us with a few questions, and we can hopefully use some of these things we talked about and build upon them.
Yeah, absolutely. That sounds great, Kevin. And that was a wonderful presentation reviewing some important concepts that will definitely play a role in the conversation and in the questions that were asked by our patients. So the first sort of chunk of questions was really around COVID antibodies, right? A lot of people are talking about getting the vaccine or have gotten the vaccine, and they don't know what to do next in terms of should they get their levels measured? Should they get these antibody tests? What do they mean? What should they do next, really? So I think I'm going to read what a couple of the patients had sent in, and then we can kind of break it down because it is a complex question. So, one patient had said, I'm on Rituximab and had the round of COVID vaccines, but my antibody test 3 weeks later showed a low level of response. What does this mean? Will there be booster options? And are you advising patients to test their antibodies after getting the vaccine? Why or why not? And I know there's a lot in there. So I do think that part of this is going to be breaking it down into sections. So what are the antibody tests actually measuring? So I think, Kevin, you did a pretty good job of explaining that, but I don't know if you want to recap just what these antibody tests are measuring so that way patients can know what we're looking at.
Sure. Generally speaking, it's going to be the spike antibody test that's being done, and the patient gets a lab report back that has a level, this concentration of the level of the spike antibody. The issue here is that we don't exactly know what level is protective. Might we in 4, 6, 12 months? Maybe, but maybe not. This is the vaccines work in a number of different ways, and it might be that if you have a stronger T cell response, you can get away with a little bit lower B cell response. So I think at this stage, my recommendation to my patients has been they can get it tested if they want. I think we have to be very careful about interpreting it, though. And I tell them that on the front end, and I kind of walk them through the different things. I mean, I do think it's really important that the patients continue, if they're immunosuppressed, continue to adhere to CDC guidelines regarding social distancing, masking, et cetera.
Yeah, we'll definitely get into some of those lifestyle things a little bit later, because there's definitely questions about that. Sebastian, what about you? What are you actually telling your patients in terms of whether to get this antibody test or not get it? And then how do you help them sort of interpret that after the fact?
So I think kind of very similar to what Kevin said, I have a very open discussion, and I kind of say the pros and cons about it. I personally haven't strongly been recommended it because I don't think it changes the message that I give my patients so far. And as Kevin pointed out, we really don't know how to interpret this. So it is very tricky at this point, and it just puts another test that we really it's probably not going to change our management. It's not going to change our recommendations. So in that case, I don't necessarily kind of go for it. I think what the important thing, and this goes a little bit with what Kevin mentioned, is that patients do need to be aware that whatever low, high, negative that comes out, that doesn't necessarily mean that they're not protected. So there's like a couple studies that I think we've probably seen with patients who don't have B cells, who don't have antibody levels, but there is a level actually of cellular response to the virus as well. This is all in vitro, this is all small, but it does reassure and kind of speaks to the point that Kevin brought up, which is antibody humoral response is just one of the ways that our body actually protects it. So kind of that's a little bit of the reassurance that I go by when talking with patients. I think the main message that I say is that the only way for someone not to be protected is if they don't receive the vaccine. That all these other kind of levels of probably we're still going to hopefully get some more information there.
Yeah, no, I think that's a great point. I'm definitely sort of in line with you guys. I haven't been really ordering it at all for my patients. I do explain sort of the pros and cons, similar to how you did. I think there's a false sense a little bit out there that this is equal to immunity against COVID. And I understand that it's definitely a marker of, an immune response, but exactly what you're saying, there's not one cutoff value where we know, hey, above this level, you're immune, and below this level, you're not immune. Because as Kevin nicely described in that presentation, there are so many different arms to the immune system that are part of our protection against this virus and any others. And so this one measure, while it does give it might give some people a false sense of security, and it might give others a false sense of really negativity and worry that they don't have any protection. And so, I think that's just a really good point, because we don't have enough information, honestly, which I know we've been saying throughout the pandemic. Unfortunately, as more questions come, we're constantly looking for more answers. But I think, I'm telling my patients that we really don't know how to interpret it. And even if it were negative or low negative, we can talk a little bit actually about some of the trials, what your thoughts are on boosters. I mean, I know that's something that's come up, and a lot of patients have asked me about it. I wish I knew a lot more about what that process is going to look like. But I don't know. Sebastian, you want to take this one first? What's your thoughts on, low levels or no response in getting a booster? I know there've been a few studies out there.
So I think it's interesting because, again, things have been done differently in different places. So, for example, just like anecdotally, in France, they've actually been given booster vaccines to either immunosuppressed patients or patients who are Rituximab or anti-B cell therapies. And they've been doing this for a while now. So hopefully we'll get some more information coming up. And actually there was one small piece about transplant patients where they actually showed that if they got a third dose that was, I want to say like several weeks after the second one, in some of them, not all of the patients, but in some of them, even in the ones who had low levels or even undetectable, there was actually an increase in the antibody levels. Again, this is all fixating in antibody levels that I think we need to kind of keep reinforcing. It's just one of the ways in which our body handles infections. There was another, so I think there was another piece, I think another small piece from Johns Hopkins that did the same with transplant patients as well. So I think it's certainly coming our way. This is something that was already kind of voiced one of the companies, I want to say, for just general population. So I think it's probably going to come the way of our patients first, and we're probably going to have to advocate and try to just prioritize our patients kind of at the beginning of the list. When does this happen? I really don't know. I think hopefully institutions are going to start kind of, again, advocating for their patients, and we can kind of get things rolling, but I really don't have any kind of plan or timeline for that to happen.
Fair enough. Yeah, I don't think any of us really do. Kevin, do you have anything to add on the idea of boosters or low dose responses?
I mean, I think it should be stated that the evidence is thin right now and that we're waiting on that. And in that context, they're not recommended. So I don't think patients should be seeking out a booster. But that being said, I really do hope the science pans out that maybe these patients, particularly the ones on medications like Rituximab or some of these immunosuppressive agents, that it really could benefit and kind of boost whatever partial response that the initial vaccine series got. So I'm very interested to look at those studies when they come out.
Yeah, me too. I think, like Sebastian mentioned, there's small studies that are there, some in the solid organ transplant group. And yeah, there, some folks who had a low level response, if they got that booster, then they sort of developed a higher level. But again, this is all fixating on this antibody, and there are T cell responses. We just don't have good tests to measure those other parts of our immune system and how active they are in battling COVID or anything else. So I do hope we get more information about boosters and what level is actually protective, and how we aim for that. But this kind of segues us into one of the other questions that was asked about timing of the vaccination with Rituximab. I know we discussed this a little bit in prior webinars or prior sessions, but folks were a little bit concerned. They wanted to get the vaccine, but they're due for the Rituximab. And some docs have obviously suggested delaying the Rituximab so they can get their vaccine. But there's still fear out there, right? The people who have had these severe presentations of vasculitis and finally got things under control on Rituximab, they don't really want to delay necessarily their treatment for fear of things acting up. So Kevin, why don't you tell me what are you kind of recommending in terms of the Rituximab timing and COVID vaccination?
Yeah, this is a very personalized decision between the patient and the doctor, because it really, a lot goes into a discussion like this. Most importantly, probably, is just how active or recently active the patient's vasculitis was. In cases like this, I believe the patients need their treatment. And I think perfect is the enemy of the good sometimes. And we can't let the systemic vasculitis that we know it can be an issue come and, sit in the driver's seat with us. So I think barring that, if the patient has been stable for some time, the most ideal time to get an injection, really of any sort. I mean, I've been saying this story for years now with flu and others, shingles, things like that, is to get the vaccine right before the patient's next dose or next series of Rituximab. You're trying to put as much time in between the Rituximab they've already gotten and the vaccine. And so, you're talking about 4 or 5, 6 months sometimes. This became a real, I lost sleep on this as vaccination became available for patients, because you'd have a patient that maybe had just gotten it, let's say December, January, and the vaccine became available kind of to patients in the several months after that. And it became very difficult to say, hold off on getting it. You don't want to be the doctor and saying that don't get it and then them get the infection itself. And so again, I had that kind of conversation with the patients that any kind of partial response might be protective. And so this is another reason why I hope the booster science pans out and boosters actually can become available, is for patients like this that really couldn't, perfect the timing, but then really do need the protection.
Oh, no, I agree. I think it's definitely an individualized decision. I think you have to work with your specific disease and your specific treating physician, because there are cases where someone's stable and doing well, and it really, I do feel it's safe to sort of delay that treatment and try to improve that vaccine response. And in other cases, their diseases just needs to be treated. And so again, there's not one sort of rote answer for everybody with that question, but yeah, that's kind of my thoughts. Sebastian, I'm going to actually switch gears a little bit, and have you take a first shot at this next question. You know, especially we just had July 4th weekend. There's masses of people everywhere. There's a lot of unmasking. There's a lot of celebrating. And this totally reasonably raises a lot of anxiety for many people, but especially our patients, right? Who are really unsure how these different mandates apply to them, what they should be doing, how should they be functioning while they're watching all of their peers kind of go back into society and revel, and hopefully, you know, turn the tide with coronavirus. How should we be guiding our patients, and what do you think our patients should be doing with all of these changes that I kind of talked about?
Well, I think first things first is everyone needs to be comfortable with what they're doing. Like it's okay wearing a mask if everyone are kind of outside. It's not. I think we need to find peace in what we feel comfortable with right now, especially given changes and uncertainties and everything else. So that's kind of the first thing. I think for probably similar to what you all have been doing with your patients is I still recommend caution based on the data that we have. I think we need to be very clear on who is higher risk or not, and a lot of our patients are. It's a fact based on kind of their medical problems, besides their actual vasculitis, the medications they're on, they are higher risk patients. We know that. So that is something that we have learned from the data so far. And that now with the kind of this concerns about protection or not, which again, such very little data, we still need to learn more about this. I think I'm still erring on the cautious side of trying to mask as much as possible, trying to avoid kind of keep circles still closed. I know it's very difficult. We've all been locked up for a year and a half, trying to be out there and see our loved ones. But I think I do recommend staying on the cautious side. And it's tricky because things are opening up, but I think we need to be a little bit more cautious on this aspect. So I'm still recommending that. I'm still wearing mine as well.
Yeah, no, that's totally fair. Kevin, do you want to add to that at all? Or what are you telling your patients in terms of social distancing, masking, and how to be in society? I mean, a lot of people work, you know, and they travel for work. Are you giving any specific guidance for what these folks should be doing?
Yeah, I think, well, that's a good place to start. I think if they are traveling, I'm telling everyone to wear a mask on the plane in the airport and things like that. I think I'm going to probably forever. And I'm probably going to wear a mask during flu season going forward, no matter what. But I think I totally agree with Sebastian. I think we've probably gotten to a point now where the masking is not as politicized and things like that. And I think folks are comfortable wearing it. They're not comfortable not wearing it. And I think our patients just need to be in that former group of, you know, if you're going to be around people that are don't be around people that are clearly ill. But if you are, please wear a mask. If you're around a lot of people that you don't know their vaccine status, wear the mask. And I would probably include the grocery stores in that. I mean, that's probably the most common sort of exposure that our patients might have in kind of a setting where they don't know what to do or not. You're going to go into a lot of grocery stores and there are going to be folks not wearing masks, but I think it's probably the thing to do. It's you know, there were viruses before this virus, and there are viruses after this virus. And I think it's just really important. I think now we just have a lot better sense of how to protect ourselves and our patients from those sorts of things. And so it's the same stuff we've been ingrained in us from the beginning of this, which is wash hands, avoid the ill, wear the mask when you're in crowded areas. I think that guidance will get you a long way.
No, I completely echo that. I know this has been an anticipated topic of discussion with a lot of great questions, and hopefully we've provided, you know, some insight and answers, although we are clearly still searching for many. Any final comments that you want to say sort of about our patients, vasculitis, vaccines, how to function?
Sure. So I think, first of all, vaccines are doing what they're supposed to be doing. We're seeing that everywhere. We're seeing that with even new variants, like the severe forms of disease, the bad cases, mortality is going down. Even in other regions of the world, which are still being very kind of heavily struck by it. So that's one. I think there are still several uncertainties. More information is coming up. Probably a lot of institutions, not only worldwide, but like certainly in the country, are doing studies. I think the Global Rheumatology Alliance is also collecting some data. Euler has collected some data. So I think besides kind of what we are all aware from the John Hopkins studies and other institutions that are slowly collecting data and giving us some more information about that, I think. And then as a quick wrap up with the topic on concern that patients ask about flares is, I think the data that is coming out is actually showing that the concern for flares, the little concern that we have for flares is probably being supported by the data. So I think it's another reason for reassurance about the vaccine and the importance for our patients to get vaccinated. So I think we'll continue to try to get data and try to share with patients as much as we can. The uncertainties that we had a year ago, we still have them, but in different topics. But the whole community is trying to move kind of in the right direction in order to keep ourselves and our patients as informed as possible.
Yeah, great. Thank you, Sebastian. Kevin, how about you? Any final sort of thoughts on or advice to our patients on vaccinations?
Yeah, I think these webinars are a wonderful source of information, and I hope that it empowers the patient to kind of take this information and open a discussion with their provider. Because I think, if anything, that's a truth in all of this, is that each case is different, each patient's different, and while we can kind of give some guideposts on behavior and approaches and things like that, I think the patient really owes it to themselves to have this conversation with the doc that's taking care of them. And, and I think, as always, I hope that patients can use one of the ways, either emailing the Vasculitis Foundation or social media, to ask questions that they want to be answered on further webinars.
Thank you. That's great. It's always a joy to have these conversations with all of you guys and be able to talk about these, tough and interesting topics. My last thoughts would really just be, get vaccinated if you're not. Wear a mask. I plan to, period, for a while. And continue to ask questions to us, to your physicians, and we will continue to try to search for answers. And hopefully this was informative. And I'll just turn it back over to Kathy. Thanks.
Well, thanks so much. I just want to say, of course, special thanks to Dr. Dua, Dr. Satui, and Dr. Byram for sharing their time and knowledge with us today. And I learned so much just in this few minutes. I have to say I was not very clear on T cells and B cells until today. So I appreciate that, Dr. Byram. And as a patient, all of this information covered the questions that were running through my mind. So I'm sure they are for everyone else. I'd like to encourage all of our viewers to visit the Vasculitis Foundation's updated COVID-19 page on the VF website. On this page, we have a new section called COVID-19 Vaccines and Antibodies. So this is where we'll be putting new articles and online resources and need-to-know information specifically dealing with this critical issue about the vaccines and antibodies and all of the questions that we discussed today. And of course, I also want to thank our sponsors, ChemoCentryx, Genentech, and Amgen, for their support of our Road to Wellness webinar series. They're helping the Vasculitis Foundation provide these valuable educational webinars and support our commitment to patient education. I always like to say also, there are some great ways to stay informed about vasculitis. On our social media, you can find us through the Vasculitis Foundation Facebook page, where you'll get the latest announcements about upcoming regional conferences, and you'll be alerted when there are new webinars and podcasts available. So like our Facebook page. Don't forget to follow the VF on Twitter at VasculitisFound. And also, one of the best ways to learn about the Vasculitis Foundation is to subscribe to the VF e-News. Every month, the VF publishes its electronic newsletter, the VF e-News, and you'll learn about current vasculitis research and open studies that are recruiting patients. You'll get links to patient profiles, updates, and the latest webinars, all posted on the VF website, and links to register for upcoming virtual conferences and hopefully one day in-person conferences again. And all you have to do is subscribe to the VF e-News under the Learn tab on the vasculitisfoundation.org website.